My beloved students, my name is Dr. Salahuddin, working as Associate Professor in Noida Institute of Engineering and Technology, Pharmacy Institute. Today, I am going to deliver a lecture on pharmacophore modeling. So, first thing, what is pharmacophore? And in this pharmacophore modeling, first we have to understand about the pharmacophore. So, what is the pharmacophore? As the general concept says about the pharmacophore is that it is made up from the two words, that is the phoros and the pharmacon. So, these two words combines together to form a phoros, that is the essential features, and the pharmacon, that is the biological activity. So, the basic structure which is required for the biological activity, that is called pharmacophore, and this concept and this original concept of the pharmacophore is first given by the Eldridge during the late 1800s. So, when we are understanding about the pharmacophore, just we have to understand about the some chemical groups or the some functional group which is responsible for the biological activity. So, in another word, we can define the pharmacophore, that the compound and the functional group which is responsible for the activity is called pharmacophore. Now, in this pharmacophore, first thing is what is the hypothesis says. This pharmacophore model is a hypothesis accounting for a observed biological activities of a set of molecule. Now, here the question arises, what is the set of molecule? Suppose we when we are working on the drug discovery, first thing we have to observe the pharmacophore for the particular type of biological activity. So, on this particular type of biological activity, we have to determine a particular set of the molecule. That particular set of the molecule combines with the target and shows its biological effects, and this model is not valid so far. So, it is able to account for differences in the biological activity of a range of molecule. So, the range of molecule, when we are having different types of molecule, so it shows the biological activity of a range of molecules. Suppose we have different compound. So, all the different types of compounds shows the different type of biological activity, and in this term we have to understand about the pharmacophore concept. Now, here every type of atom or group in a molecule, as we know that in a molecule we are having a atom or group, exhibit a certain properties. Exhibit a certain properties means either it is act as anion, either it is cation, hydrophobicity, or it will act as a hydrogen bond acceptor, or it will hydrogen bond donor. So, the every type of atom or group which exhibits certain properties. So, this type of properties shows a different type of feature in a molecule. So, in the molecular recognition, it can be reduced to a pharmacophoric feature. Suppose if we have a molecule with certain type of atom or group. So, in that particular certain types of atom or group, we have to determine the different type of properties related with the atom or group. And next thing, this molecular pattern can be labeled as hydrogen bond donor, acceptor, cation, anion, aromatic, hydrophobic, and any possible combination. So, if we are having a molecule which are having the aromatic group or which are having the some electron withdrawing group or electron donating group or some other functional group. So, these are combinedly called this properties related with this hydrogen bond donor, hydrogen bond acceptor, cationic properties, anionic properties, aromatic properties and the hydrophobic properties. Now, here in the pharmacophore modeling. So, first thing, in a set of molecule, suppose we are having a set of molecule. So, in this set of molecule we are able to design a pharmacophore. So, in a set of molecule, the common feature of all these molecule combined together to get the pharmacophore. So, in this pharmacophore we have to identify the basic features of all the compounds. So, here we are taking the molecule and in this molecule we are having atom without pharmacophoric features, and after the creating the model of this molecule we are having a different type of colors, and after the creating the model we are having this hydrophobic model, hydrophobic properties, aromatic properties, hydrogen bonding acceptor properties, hydrogen bonding donor properties and the ring atom. So, what basically happens when we are creating the model? After the creating the model, this hydrophobic, aromatic, hydrogen bond donor, acceptor, these all the properties will performs the activity. So, these will responsible for what? These will responsible for biological activity. So, this is the example of the pharmacophore, and here we are having some ring system. After compressing the ring system, we develop this pharmacophore model. So, this figure shows an example of creating the pharmacophore model from a single molecule atom and converted into nodes and level with the pharmacophoric features in terms of this terms of hydrophobic, aromatic, hydrogen bond donor, hydrogen bond acceptor. Now, in the pharmacophoric concept in the computer aided drug design. So, the basic concept, the pharmacophoric concept in the computer aided drug design. So, as I have already discussed about the every type of atom or group in a molecule exhibit certain properties and the molecular recognition, and after combining all these properties with the molecule we can call it as the pharmacophoric feature. In the pharmacophoric features, the most common pharmacophoric features in the molecular pattern we can label as this hydrogen bond donor or acceptor, and the cationic features of the molecule, if the molecule having any positive charge, anionic features of the molecule, the aromatic group present in the molecule required for the lipophilicity, and hydrophobic, and if there is any possible combination of the properties related with this pharmacophore we can take in. Now, in the pharmacophore fingerprint. So, in the pharmacophore fingerprint is nothing but it is a collection of the pharmacophoric features in the 3 point or a 4 point set of pharmacophore generated for each ligand. So, the pharmacophore fingerprint is an extension of this concept and typically annotates a molecule as a unique data string. So, in the unique data string in the 4 point in the 3 point or the 4 point set of pharmacophoric features, we have to identify the features present in the ligand. in the form of hydrogen bonding acceptor, hydrogen bonding donor, cation, anion and so on. Okay. So, we are going to calculate the distance between this between the feature point. Feature point is the characteristic points present in the molecule, present in the atom, present in the group. So, here the distance between the feature point is counted in a bond for topological fingerprint or the distance banding when using 3D fingerprint. Now, here in this pharmacophore fingerprint, here we have the 4 figure A, B, C, D and 5 figure. So, here what is A? What is A? The pharmacophore fingerprint is the representation of the small molecule that is ligand. So, here we are using this small molecule, molecular weight is less than 500. So, here when the molecule is small, we can represent in the pharmacophoric fingerprint. In the next thing, molecular interaction feature. What type of the feature is present in the molecule which is required for the molecular interaction with the target. So, that we have to identify the molecular interaction features in the ligand. Like this, the different type of the color shows the different type of features. Here the hydrogen bond acceptor, the cationic and hydrophobic, hydrogen bond donor, anionic and the aromatic. This different type of color shows the different type of feature in a molecular as the molecular interaction features. Now, come to figure C. This is the possible 3-point or 4-point combination of the molecular interaction features. So, not all the features is responsible for the interaction, only the 3-point or the 4-point interactions feature is responsible for the interactions with the target. Now, come to the D. Here the different distances between the features calculated either through space or by a number of bond length. We have to calculate the distances between the features present in the ligand, or by calculating this different type of feature present in the ligand, we are able to identify the different types of the features here. Now, this the last one is the frequency of occurrence is stored in a string. Now, this model, this is the pharmacophore model or query. If we are having a pharmacophore model, so these different type of clue like hydrogen bond acceptor, donor, anionic, cationic, aromatic. So, it consists of a few features organized in a specific 3D pattern. So, in these 3D pattern, due to the presence of due to organization in the form of 3D, not all the features is responsible, only 3-point or 4-point features is responsible for the pharmacophoric features only. So, each feature is typically represented as a sphere in the form of a sphere. So, in the form of a sphere, there is a various variant exist with the radius determining the tolerance on the deviation on the from the exact position. So, here the different type of spheres have different types of characteristics present in the ligand. Now, here this pharmacophoric model consists of the few features organized in a specific 3D pattern. Each feature is typically represented as a sphere, and the feature can be labeled as single feature or any logic combination of and or not to combine the different interaction pattern within the label. So, here additional feature can be described forbidden volume interactions typically to represent the receptor boundary. This one. Now, come to the pharmacophoric modeling. What are the different type of pharmacophore modeling we can done in the computer aided drug design. So, first thing is ligand based pharmacophore modeling, and the next one is structure based pharmacophore modeling. Here I am showing the one ligand, and here there is a one receptor. So, receptor, the structure of the receptor. So, the pharmacophore modeling is of two types, that is the ligand based pharmacophore modeling, that is also called indirect drug design, indirect pharmacophore modeling, or the another one is structure based pharmacophore modeling, or we can say it is a direct pharmacophore modeling. So, in the ligand based pharmacophore modeling, we can do the virtual screening, we can do the lead optimization, and in the structure based we can do this de novo drug design, means beginning of the drug designing and the target identification. Now, in the structure based pharmacophore modeling, first I will discuss about the structure based, and here in the structure based these are the various steps involved in the structure based pharmacophore modeling. So, first thing is examination of the 3D structure of the biological target. It can be done with the help of X-ray crystallography or NMR spectroscopy. So, we have to examine the 3D structure of the target with the help of X-ray crystallography and the NMR spectroscopy. After doing this, identification of active sites. Identification of the active sites is what the different type of site, different types of groups, different types of functional group which is responsible for the activity we have to identify, that is called identification of the active sites. Now, next is evaluation of the structure. So, evaluation of the structure after the identification of the active sites, which type of the active site and basically what is happening in this active sites, these are done with the help of evaluation of the structure. Now, after the evaluation of the structure, we have to synthesize the drug candidate against this particular structure. So, we are not synthesizing any type of drug candidate, we have to synthesize the specific drug candidate which is identical to its structure. So, after the synthesis of the drug candidate, we can perform the biological screening of all the synthesized drug candidate, and then we will get the lead compound, and after the lead compound we can do the lead optimization. This is very important step in the computer aided drug design. So, here lead compound discovery. Next thing is pharmacophore modeling in the virtual screening. So, the pharmacophore modeling in virtual screening, that is what we can do with the help of this pharmacophore modeling in the virtual screening. So, first thing what we are doing in the virtual screening. So, in the virtual screening, screening of the compounds, in order to identify the molecule to the desired biological effects. Whatever the desired biological effects, that we can do with the help of this virtual screening. We are screen n number of ligands, screening of the structures with the help of this virtual screening. So, for this purpose researcher create a 3D representation, the 3D structure of the pharmacophore, that is called query, that most likely encodes the correct 3D organization of the required interaction pattern. So, we have for the this virtual screening, we have to divide the data into two sets. The first set is the training set and the next set is evaluation set to validate the generated pharmacophore. Whatever the pharmacophore that we have generated with the help of this virtual screening, then when we multiply the active ligands and inactive derivatives are known. Here this pharmacophore modeling in the virtual screening, we have 4 boxes. So, here we have this active ligands, the known active ligands, either it is known or not. If it is no active ligands and the no protein structure. Now, come to the second, the ligand is known, but the structure of the target is not known. Now, the third one is ligand is not known and the structure of the target is known. Fourth condition is the ligand is also known and the target is also known. So, here in the virtual screening there are 4 possibilities that we can do the virtual screening and we can get a pharmacophore model, that is no protein structure and the no ligand structure is known, no protein structure, but active ligand structure are known, protein and ligand structure both are known and the last case is only the protein structure is known. Now, in the pharmacophore methods in docking, what is the role of this pharmacophore modeling or the pharmacophore methods in the docking simulation? As we know that in the docking we are doing the database of the ligands that we can filter using the pharmacophore query. The pharmacophore query that we have already obtained from the virtual screening prior to the evaluation using the docking simulation. So, in the docking simulation it can be post filtered using the pharmacophore query to remove any compound that failed to bind according to the pharmacophore query, and the ligand are evaluated in the absolute conformation and should not be allowed to align without with the pharmacophore feature. So, here this pharmacophore modeling we can apply in the docking simulations. So, another alternative of the pharmacophore alignment to guide the placement during the docking simulation. So, the pharmacophore is required, pharmacophoric model is required in the docking simulation. So, with the help of this pharmacophore model, we can identify the we can choose the target and perform our docking simulation. Similarly, to the fitting of the molecule into the pharmacophore query or to guide the placement by using the constraint while scoring the different docking poses. These are the some references and that we can observe the pharmacophore modeling and the pharmacophore modeling with the help of the ligands that we can do by observing the some important features present in the ligand, and we can perform this in the virtual screening, in the docking simulation and so on. Thank you so much.